We are group EBH, and today we'd like to share with you how we attempted to characterize our unknown gene in Brachypodium distachyon. So to begin our research, we started by generating an ab initio and a homology-based model for our gene in order to get a sense of the major transcriptional and translational landmarks within our gene sequence. The FGANESH algorithm predicted that our gene was encoded on the positive DNA strand, contained 7 exons, and likely encoded a polypeptide of about 330 amino acids. Our homology-based model from phytosome predicted very similar landmarks, particularly in terms of the location and length of exons in our gene sequence, and also identified our gene as Brady1G68250. And I would also like to note that the RNA log scale coverage from phytosome showed elevated transcript abundance in regions corresponding to the identified exons based on the existing cDNA library, which strongly suggested that our identified exons from these models were pretty accurate. Overall, these similarities gave us confidence about the characteristics of the EBH gene sequence, as well as the fact that our gene is expressed in Brachypodium distachyon. So next, we conducted a side-blast of our gene's coding sequence in the NCBI conserved domains database. And as you can see here, our top domain hits were all related to superfamilies of GTPases, suggesting that our gene is likely a GTP binding protein, or more specifically, a small GTPase. This was interesting to us because GTPases are implicated in a variety of cellular pathways, such as cell signaling and membrane trafficking, and the role is often to function as biochemical switches that turn off or on pathways by binding and hydrolyzing GTP. The side-blast annotated our gene as an isoform of the small GTPase LIP1 in Arabidopsis thaliana, which we found is a protein involved in circadian rhythm regulation. So here I would like to highlight some of our results in a paper from 2007 that was referenced in the side-blast annotation. And in one experiment, they observed how a loss of function mutation, or delta LIP1, would affect the oscillation period of the plant circadian rhythm based on the luminescence of circadian rhythm reporter genes. And they found that during the light-sensitive period, the shortening of the oscillation period did not occur in the mutant as it did in the wild type. So as such, they concluded that LIP1 was involved in circadian clock regulation, as well as entrainment or the resetting of the circadian clock. And in another paper, we found that the LIP1 protein in Arabidopsis is involved in salt stress tolerance. As you can see on the figures to the right, we see that germination and root length in the mutant plants with the loss of function mutation in LIP1 were more suppressed under increasing salt concentrations compared to wild type. However, when we generated a phylogenetic tree of our gene, we did not see the LIP1 small GTPase from Arabidopsis. However, we did see other GDP binding proteins from other related plant species. And so we concluded that the GTP binding domains of our gene were well conserved across these related plant species, most of which were also small GTPase LIP1 isoforms or various Rab GTPases and other GTP binding proteins.
So when we looked at the thermocycler data for our gene, we saw that there was just absolutely no correlation between the relative expression of the gene and either the thermal or the photo conditions. So clearly light and dark or hot and cold conditions are unrelated to the expression of the gene EBH. So we were able to effectively rule out circadian rhythm regulation as a possible function of the gene. And so then when we also looked at the Brachypodium EFP browser to see where expression was highest of the gene, we noticed it was high in roots, old internodes, and young leaves. And so some theories that we had based off of that is that our our gene could be involved in plant development since the expression was higher in the young leaf compared to the old leaf. Alternatively, it could be involved in nutrient uptake because it is highly expressed in the roots in the old internode. And of course, nutrient uptake happens in the roots, and the old internode is where nutrients and water pass through to get to certain structures in the plant. And so then when we performed statistical analysis, we saw that the expression of the gene was high in root, stem, and leaf, and the differences in expression between the roots, stems, and the leaves was statistically significant. So the function of the gene could be affected by the different structures. And so when we designed our qPCR experiment, since we saw that was so high in roots, we decided to base it in the roots. And so since we were able to rule out circadian rhythm regulation, we decided not to test that at all, but rather focus on the salt stress tolerance. And so our hypothesis was that expression of our gene would increase in roots after we incubated the roots for 3 hours on a 200 millimolar sodium chloride growth medium. So our overall experimental design was to grow 40 seeds for 7 days on a growth medium and then transfer the seedlings to either a control or a treatment medium, the control medium just being a normal growth medium, while the treatment medium was a 200 millimolar sodium chloride medium. And so then after 3 hours of leaving the seedlings in these new conditions, we harvested the roots and extracted and quantified the RNA. Ultimately, the RNA in the salt-treated roots was very low, so low that we didn't actually have sufficient data to even analyze the effect that the salt had on the expression of the gene. So we ended up just looking at relative expression levels of our gene in the wild type versus the mutant plants. So even though there was a generally higher level expression in the mutant plants, it wasn't a statistically significant difference. So overall conclusions from the semester and from our experimental design was that our gene is not regulated by the photo or the thermocycler. So again, not related to circadian rhythm regulation. It's possible that 200 millimolars of sodium chloride was lethal since we had very low RNA in those sodium chloride treated roots, but we also weren't sure if perhaps we just hadn't extracted the RNA well enough. So in the future, if someone were to take a look at our gene and possible functions, redoing that experiment in the qPCR with lower concentrations of salt would probably be helpful and might give a more clear result to salt stress tolerance and whether our gene is involved with that. We also do have our theories based on which plant structures the gene was highly expressed in. So it's possible, again, that the gene is involved in plant development or nutrient uptake, but we have absolutely no literature or experimental support for that. So future experiments could certainly take a look at it. So our acknowledgements, we would like to give a special thanks to Dr. Hazen, Dr. Dorfman, Nishadi, and all of our classmates for supporting us throughout the semester. Thank you so much.